Okay, we're going to make a start now. So welcome everyone to our A Level AQA Biology session. In today's lesson, we are going to be looking at the light independent reaction or the Calvin cycle. So we've already looked at the light dependent reaction, the LDR, and that was sort of a couple of weeks back. Remember, we're switching between year 13 and year 12 topics, so it's beneficial for everyone. Today's lesson, we're going to focus on the light independent reaction, the Calvin cycle. We're going to be looking at essay points that basically a method or a model 6 mark answer that you can learn and write the same exact thing in an essay question. We're going to be looking at that as well. We're going to go through the process, and then we're going to be looking at a couple of exam questions towards the end of the lesson. As always, if you have any questions, you can let me know in the Q&A, and I'll be more than happy to answer them for you. So if we make a start now, so remember we're looking at photosynthesis, and we looked at the process of the light dependent reaction in our last lesson on photosynthesis, and that was this first part of the spec. Now, the second part of the spec from here to here, that's literally just talking about the light independent reaction, and it's going through the method for the light independent reaction, which we're going to be looking at in today's lesson. So we looked at an overview of photosynthesis previously, and we're just going to recap that quickly right now. So photosynthesis, we talked about it being a two-stage reaction. It was a two-stage reaction that consists of two stages, which is the LDR, the light dependent reaction, or the LIR, which is the light independent reaction. Those were the two stages of photosynthesis that we spoke about previously. Okay, and we've already looked at the light dependent reaction. And the key difference that you also have to acknowledge is that in these two stages, you have the light dependent reaction and the light independent reaction. They occur in two different locations. They both occur in the chloroplast of the plant cell, but they occur in two distinct different locations within the cell. So the light dependent reaction, which we looked at previously, occurs in the thylakoid membrane or the grana of the chloroplast. The light independent reaction, which we're looking at in today's lesson, it occurs within the stroma. Okay, that's where the light independent reaction occurs. It occurs within the stroma of the chloroplast. So we saw that the LDR, the light dependent reaction, was the first stage of photosynthesis, and it requires light. It is dependent on light. And then whatever products which are formed from that reaction, it is then used in the next stage of photosynthesis. It is used in the light independent reaction or the Calvin cycle. So the light energy and water that is used in your light dependent reaction, it is used to create ATP and reduced NADP. Remember when we spoke about reduced NADP? Reduced NADP just means NADP which has gained a hydrogen. So it's NADPH. And remember the P because in here in respiration there is no P, it's NAD. So we have NADPH. Okay, and both of these products, ATP and NADPH, they are both needed from this reaction for the light independent stage, the light independent reaction of photosynthesis, which is occurring in your stroma. So the key takeaways, obviously this was just an overview of the whole process of photosynthesis, but the key takeaways that you need from this slide is the light independent reaction occurs in the stroma. That's a common exam question. It occurs in the stroma. Okay, it's the second stage of photosynthesis. It doesn't require light. However, it uses the products which are formed during your first stage, which is your ATP and reduced NADP, and it uses it for the second stage. Okay, so we're now going to move on to the Calvin cycle. Now you can refer to it as the light independent reaction or the Calvin cycle, whichever one that you prefer. However, I'm going to be referring to it most of the time as the Calvin cycle. Now, the Calvin cycle occurs within the stroma. And these are just key points. We've not actually got on to the method. We're just looking at key points. Okay, we're going to be looking at the method on the next couple of slides. So the Calvin cycle occurs within the stroma, and there is an important enzyme. Okay, the fluid, which is the stroma, it contains this specific enzyme, and this specific enzyme is called rubisco. And the purpose of an enzyme, remember, an enzyme is a biological catalyst. The enzyme, in this case, rubisco, is going to catalyze this reaction that's going to take place. And because we're using enzymes, we're using the rubisco enzyme, we know that enzymes are temperature sensitive. If they are temperature sensitive, that means at high temperatures, they will denature. Okay, so we don't want the temperature to be too high, or photosynthesis won't be occur because this enzyme of rubisco will denature. Okay, so that's the first key important point that you need to be aware of. It occurs in the stroma and it contains this special enzyme, which is rubisco, which catalyzes the reaction. Because we're using an enzyme, this stage of photosynthesis is temperature sensitive. Now, the Calvin cycle, this is what it uses. It uses carbon dioxide, it uses reduced NADP and ATP from your light-dependent reaction, and it does this to form a hexose sugar. Now, what does a hexose sugar mean? It means glucose or some respiratory substrate. A hexose sugar just means that it's made out of 6 molecules, okay, 6 parts. And these 6 parts will then be used as an energy reserve within the plant. Okay, so it's important to acknowledge the reactants of this stage. You've got carbon dioxide. Now, carbon dioxide, remember when we looked at the reaction of photosynthesis, we know that carbon dioxide is reacting with water, and it's forming your glucose and your oxygen. Now we've seen the water already being used. This was used in your light dependent reaction. And now we need the carbon dioxide. Where are we going to get this carbon dioxide from? This carbon dioxide literally comes in from the Calvin cycle. So the second stage of the reaction, the second stage of photosynthesis requires carbon dioxide. And that's where the carbon dioxide comes into play. Okay? So this is in the second stage or the Calvin cycle of photosynthesis. And the purpose of this is using carbon dioxide, which has freshly entered this leaf in this chloroplast, and it's using the products from the previous reaction, NADPH, ATP, and it's combining everything and it's forming a hexose sugar, it's forming glucose. Oxygen was produced in the first stage of your reaction. Glucose is being produced now. Okay, glucose is going to be produced now in the second stage of your reaction where you're forming a hexose sugar. The ATP, which was produced previously, is hydrolyzed to provide energy for this reaction. So that's where the ATP. So we know that reduced NADP is required. We know that ATP is required. Where does the ATP go? The ATP is an energy source and it is used in this reaction because this part of the reaction requires energy. That's why we need ATP for this part of the reaction. And then finally, that's where the ATP is going. ATP is used as an energy reserve. We need ATP for energy in the plant for photosynthesis to occur. Okay. Reduced NADP, the NADPH that is produced, that is used to reduce GP molecules in the cycle. Okay, so again, this will make more sense, but the NADP is used because it has a hydrogen. Remember, it's not just NADP, it's NADPH because it's being reduced in the first stage of photosynthesis. And because this NADP has a hydrogen, that's what makes it useful. It can reduce other things, it can break other things apart. So this hydrogen is then donated to this GP molecule, and then that GP molecule is broken apart. Okay, so that's what's happened. So again, from this slide, the key takeaways from this slide, which you need to be aware of, is that, again, we've already spoken about the Calvin cycle taking place in the stroma. There's an enzyme which is called rubisco, and that's the catalyzing part of this reaction. The Calvin cycle, it's going to be using carbon dioxide because that's an important part of photosynthesis, and you're going to be using the products from the previous reaction, which is your NADPH and your ATP. And the overall aim from this reaction is to produce your not your oxygen, the glucose that is at the products of this reaction. The ATP from the previous reaction is used to release the energy required for this reaction, and the reduced NADP that we formed from the previous reaction is used to reduce the GP molecules which are within this cycle. It's to donate the hydrogen that is within this cycle. Okay, so that's the key points that you need to be aware of. We're now going to be looking at the Calvin cycle itself. The biggest area of the spec is just learning this Calvin cycle, as a lot of questions end up coming on it. Now, this is the Calvin cycle, sort of in image form. Now, I'm going to constantly be referring to this image again and again, but we're going to be looking at the method first. So the first stage, okay, the CO2 from the atmosphere enters and reacts with ribulose bisphosphate. Now, that can be shortened to RUBP. You don't need to learn the full word. Okay, you just need to learn RUBP. That's the form. So what's going to happen is that the CO2 is going to enter and it's going to react with RUBP, and it's going to form two molecules of glycerol 3 phosphate. Now, glycerol 3 phosphate, again, it's not a word you need to know. You can just learn GP. And the reaction is catalyzed by the enzyme rubisco. And I'll flick back to the other slide. But if we think about it, what's happening is that CO2 is coming in from the atmosphere. It is reacting with RUBP, so RUBP, which is found within the stroma of the chloroplast. And when it reacts with RUBP, it's going to form two lots of GP, okay, two glycerol phosphate molecules. Okay, and this reaction is very slow to start off with. So to speed this reaction up, we use the enzyme rubisco. Rubisco enzyme is used to catalyze this reaction. Now, instead of learning this worded answer, if you just learn this, you can easily work out what a worded answer would be in an exam. You can say that CO2 reacts with RUBP catalyzed by rubisco to form two molecules of GP. You can literally work that out from this one equation that I've written down here, instead of learning that whole answer. Okay, so let's flick back to the previous slide over here. As we can see over here, what's happening is that we have RUBP, and RUBP is a 6-carbon molecule. It contains 6 carbon atoms. What's going to happen is that CO2 is going to enter the stroma, and that CO2 is going to react with this RUBP. And when it reacts with this RUBP, it's going to be catalyzed by the enzyme rubisco, and it's going to form two molecules of GP. Now, two molecules of GP, all that means is that RUBP has been broken apart. Okay, it's been broken apart into, sorry, this is a 5-carbon RUBP. RUBP is 5 carbon. Okay, it reacts with CO2 because CO2 has 1 carbon. And what's going to happen? 5 carbons add 1 carbon, it produces 6 carbons, and it forms 2 different molecules of GP. You have 1 over here and 2 over here. Okay. So as you can see, that's the first stage of the Calvin cycle. Just remember that equation that we went through. RUBP is going to combine with CO2, catalyzed by rubisco to form 2 molecules of GP. That's all you need for this stage of the reaction. RUBP is a 5-carbon molecule. As you can see, it's 5 carbons, and CO2 is a 1-carbon molecule because there's 1 carbon in CO2. And what's going to happen? 1 carbon combines with 5 carbons to form 6 carbons. 6 carbons is split apart to form 2 molecules, 3 carbons each. Okay, so CO2 reacts with RUBP to form 2 molecules of GP, and that has been catalyzed by the enzyme rubisco. Now, the second stage of this reaction It says GP is reduced to triose phosphate. Now, because GP is reduced to triose phosphate, this is obviously going to require energy, and this is where the ATP comes into play. It uses the energy from ATP, and it accepts the hydrogen from reduced NADP. Okay, so obviously GP, we formed two molecules of GP. That GP now has to be converted into something else. So in this reaction, what's happening is the GP which is formed, it is being converted into triose phosphate. Now you can't abbreviate this to TP. You have to say triose phosphate. You have to say the full name itself. You can't abbreviate this name. So GP is reduced into triose phosphate. How is it reduced? ATP is used. And what else? You've got the H plus from NADP. So the NADP, it fulfills its role here. It gives the hydrogen to GP to reduce it into triose phosphate. Okay, so again, it's much simpler than learning the worded answer to think of it as chemical equations. CO2 combines with RuBP to form two molecules of GP catalyzed by Rubisco. The GP is then reduced into triose phosphate using ATP and reduced NADP. Okay, and the hydrogen from reduced NADP is literally donated to the GP to turn it into triose phosphate. So if we look back at the image over here, we've got two molecules of GP. Now, what happens to these two molecules of GP is that it is converted into two molecules of triose phosphate. It is converted into two molecules of triose phosphate using ATP and reduced NADP. Okay, that's basically what's happening each time. ATP and reduced NADP are being used to form two molecules of triose phosphate. I know it says TP there, but ignore that. You just need triose phosphate. Okay, and then in each turn of the cycle, what's happening is that one carbon is leaving. One carbon is leaving and it's being collected to form useful organic substances. Now, the reason why it doesn't just say glucose is because useful organic substances, it can form trisaccharides, disaccharides, all that sort of such as well. Okay, it's not specifically one thing that it forms. Okay, so at each turn, one carbon leaves the cycle. And when one carbon leaves the cycle, it's converted into useful organic substances. And then once you form 6 turns of the cycle, you form a molecule with 6 carbon atoms. And that is a hexose sugar, which is used. Okay, so if we have a look at this diagram over here, we formed our triose phosphate. Now, at the end of each triose phosphate, what's going to happen is that one molecule, one carbon atom is going to be released. One carbon atom is going to come out and it's going to be picked up. So over here, we had a 6 carbon atom. Okay, one carbon atom is going to be released. So now you're going to have 5 carbons left. And then those 5 carbons are going to use ATP to reform this RuBP that we originally had. And then the cycle keeps on repeating in circles. It keeps on going on and on and on in a cycle. So at each cycle, so you've got CO2 combining with RuBP to form 2 molecules of GP using the enzyme of Rubisco. Once you've formed 2 molecules of GP, the GP is then reduced into triose phosphate, 2 molecules of triose phosphate, and it does this by using ATP and reduced NADP. Once your triose phosphate has been formed, one carbon atom is removed, so you have 5 carbon atoms left over, and those 5 carbon atoms use ATP to form RuBP. Okay, so those 5 carbon atoms, they use the ATP to reform the RuBP. And once your RuBP is formed once again, the cycle can continue going round and round once again. Okay, so that's basically the whole process of the Calvin cycle. Okay, I think there's one more step. Yeah, the rest of the molecule is used to regenerate RUBP with energy from ATP. That's what we've just looked at. Okay. Now we will have a look at a polish method, and I think there's an exam question with a model mock scheme method that we're also going to look at. At the moment, just get a grasp of what this method actually looks like, what the process actually is. Just get around some understanding the process. Okay. So additional key information, that's what we're going to move on to now. We've looked at the Calvin cycle. Again, there are some key points that you need to be aware of. Okay, and that's what we're going to be looking at now. So additional key information. Okay, so in terms of additional key information, glucose is the product of the reaction. However, this can go on to form other molecules such as disaccharides or polysaccharides or trisaccharides or anything like that. Glucose is formed, however, then that glucose can then go on and it can form different sorts of molecules. That's why we have to say that useful organic substances are formed from this process of photosynthesis. It's not just that it's not just that glucose is formed. Glucose is formed, however, then that glucose can go and it can form different sort of disaccharides, polysaccharides. That's why we just use the term useful organic substances. Okay, organic means containing carbon. Now the next point is temperature. Now temperature affects this stage of photosynthesis. It says LDR. It affects the LDR stage and it affects this stage because in the LDR stage we have ATP synthase, and in this stage we have Rubisco. Because we have Rubisco, both of them are temperature sensitive. And because both of them are temperature sensitive, they can denature the enzymes. And then the concentration of CO2. Okay, this is an important one. We didn't need CO2 in the first reaction, in the light dependent reaction, but we need CO2 in the second reaction, the light independent reaction or the Calvin cycle. Okay, and the reason why we need CO2 is because the CO2 needs to combine with RUBP. If we don't have CO2, then nothing will be able to combine with RUBP. So we need CO2 to combine with RUBP, or you wouldn't have any sort of reactants within your reaction. We need reactants for a chemical reaction to occur. Okay, so that's the additional key information that you need to be aware of. We're now going to be moving on to the essay points, okay? The essay key points that you are required to know. And think of it, when you go into an exam and you're writing an essay, you basically have to learn model mock scheme answers. And if you learn model mock scheme answers, you literally just have to write that model mock scheme answer and focus on the aspect the essay is basically telling you to. So the more model mock scheme answers that you learn for processes such as this, the stronger your essay writing skills will become. So the essay key points, I've literally summarized the method over here. This is the mock scheme method that you need to know for the Calvin cycle. CO2 reacts with RUBP, catalyzed by Rubisco, forming two molecules of GP. GP is then reduced to triose phosphate using the products from the light-dependent reaction NADPH and ATP. Some triose phosphate is converted into useful organic substances such as glucose, and some triose phosphate is used to regenerate RUBP using the energy from ATP. So as you can see, we've literally written out the method. Okay, we've written out the method, which is all correct. And whenever a question comes, this is again a common topic which many people use to include within their essays. The light dependent reaction is one because it's a long process, and another long process that you have is your light independent reaction or the Calvin cycle. Now, some areas where you can include it, I put some examples. Questions from essays that you can be asked: the importance of proteins, the importance of cycles, the importance of ATP, the importance of energy transfers within and between the environment, the importance of carbon, the importance of enzymes. All of these questions, you can literally include this model answer because proteins, we've got Rubisco. Rubisco is an enzyme, and as goes for enzymes, and enzymes are proteins basically. Okay. Importance of cycle, Calvin's cycle. It's a cycle, so you can include it. Importance of ATP, ATP is used in the stage. Importance of energy transfer, energy is being transferred in photosynthesis, so you can definitely include it in an answer like this. And carbon, you've got your glucose, you've got CO2, all that is carbon, so you can definitely include it as an answer. I don't think the importance of carbon has been on for quite a while. So again, that's a common question which could come up. So those are the essay points. That's the modern method that you can include within your answer.